Mangrove ecosystems around the world are an essential component of a healthy ocean. The unique population of mangroves in South Australia's gulfs act as nurseries and feeding grounds for common commercial fish species such as King George whiting, yellowfin whiting, blue swimmer crabs and king prawns. They also serve as a habitat for migratory and local birds, and their productivity flows onto supporting offshore ecosystems. Many regional communities also benefit from mangroves as they support recreational and commercial fish stocks, which in turn provides food, creates jobs and attracts tourism. As climate change worsens, mangroves, like every other ecosystem, are threatened. The economic and ecological importance of mangrove systems makes them a worthwhile asset for the investment of money, time and research. Mangroves exist where land meets the sea, in the intertidal zone. The mangroves in South Australia are unique due to the intertidal zone expanding up to a massive 2 kilometres. This is the water level at near high tide. And this is 6 hours later at low tide. The tide won't return for another 6 hours. As the tide recedes, pools of water remain in the creeks, trapping fish. Regular extreme summer temperatures combined with the extended low tide periods result in the remaining pools of water warming up and developing conditions predicted for the future. Nearshore environments are predicted to more frequently experience extreme water conditions with high temperatures, high acidity, high salinity and low oxygen. These conditions impact the ability of an organism to maintain homeostasis. Just like humans are sensitive to their environment, so are fish and crustaceans. Utilising these mangroves where extreme conditions occur naturally provides us with a natural laboratory to test how climate change may affect the fish and crustacean communities living here. With the help of volunteers, we sampled 4 locations, each with 4 sites, on 4 occasions over the 2022-23 summer. At low tide and during the warmest part of the day, we deployed temperature, oxygen and pH loggers in the pools, partnered with GoPro cameras. A salinity test was taken before moving on to deploy at the next 3 sites. The cameras are left to record for 2 hours, with another salinity sample taken at the 1 and 2 hour mark. This was repeated for each location over a 4-day window to minimise variance in weather conditions. A 5th visit to each location was undertaken to net each site for long-finned gobies and small-mouthed hardyheads. Fish were caught, humanely killed using the ikijimi method, measured, weighed and placed on dry ice for dissection in the lab. These species occur across all sites and locations, making them ideal indicator species. The main finding of the study was unexpected. Here we can see that with increased temperature, the diversity found in the mangrove creeks also increased. This indicates a high level of resistance the fish and crustaceans here have against extreme water conditions. This is promising for the future, as these communities may be pre-adapted to future conditions as climate change continues. However, these communities may also be at their thermal threshold, where further warming may push them past the maximum temperatures they can withstand. If this is the case, more sensitive species to disappear from these mangrove environments, upsetting the balance of food webs and having a domino effect onto offshore marine systems. Local fisheries and regional fishing tourism will also suffer as a result. The other water conditions that were measured, pH, oxygen and salinity, did not appear to have any significant influence on the mangrove communities, indicating tolerance to the conditions present at the time of sampling. The results from the fish netted showed no significant differences in their Fulton's Health Condition Index. This means that both of the indicator species maintain similar health across all sampled locations and sites, despite the differences in water conditions. Their high tolerance to varying water conditions is promising for adaptation to the future. Future studies looking at the thermal tolerances of commercial species and indicator species are required to continue answering the question of how climate change may impact mangrove communities. Annual replication of these methods would also be informative to see the variance between different years and to see if other patterns emerge.